RNA in some waters which are from some lakes from British Columbia. He's going to tell us about whether that works or not. So this is to do this prebiotic chemistry.
Yep. Sorry, give me 1 second to get set up here. Okay. If you're on Zoom, hopefully you are seeing that and you can hear me. Yeah, so thanks, David. I'm Zack Cohen, like David said, from the chemistry department. And I'm really excited actually to be here telling you guys about this work that I did last fall during my research rotation, where we investigated RNA copying in carbonate lakes. And that could have implications and possibly enable the origin of life. So I just want to start for one slide to sort of motivate. Yeah.
Can you exit out and share your full desktop and then
Sure thing.
play it? They're only seeing like half of the slides.
Oh, interesting.
Yeah. Okay, looks good. Thanks.
Okay, great. Yeah, so why research the origin of life? I would say that the origin of life informs our search for life elsewhere in the universe. Get rid of this. So yeah, so if you want to know, can we detect life on some particular planet? It depends a lot on whether life can originate there. So the origin of life in general is a very difficult problem to solve. So it makes a lot of sense to start with the origin of life on Earth. So let's talk about life on Earth. For the most part on Earth today, life is made of cells. So our bodies are composed of trillions of different cells, different cell types. And then on the other hand, you've got, in microbial world, single-celled organisms. The one exception to this rule that I guess we should mention are viruses, which are, debatably alive. But for the most part, we can go with this generalization that life on Earth is made of cells. Sorry, I'm going to try and move this thing from the top. Okay, but maybe it's not going to work. Yeah, so okay, so life on Earth is made of cells. What's a cell? I have just a very simplified view of a cell, which is just a bag of chemicals. So I would say that this model for cells, we have 3 key components. The first is a membrane. So this forms the boundary of the cell. You could think about it as like a bag that keeps the other chemical components on the inside. We also have DNA, which stores the information needed to replicate the cell. And we also have proteins, which then enact that replication. So that was modern cells. The first cells on Earth may have used different components. So in at least, the hypothesis that I'm telling you about, one of those components is still a membrane, but unlike modern cells, the membranes of the first cells could have been composed of environmentally available molecules, specifically fatty acids. So some work I've done previously is to categorize and try to determine what the sources of those fatty acids could have been on the early Earth. And there are a couple, from meteorite delivery to surface to the synthesis on hot metal surfaces after an impactor, or like Miller-Urey type electrochemical sparking. So there are sources for these membrane-forming molecules on the early Earth. The second component of the first cells could have been RNA. So just like DNA, RNA stores the information that it needs to replicate itself in its sequence of A, G, C, and U for RNA instead of T. So that's RNA as an example of RNA playing the role of DNA as information storage. But an RNA can actually play the role of protein as well for the first cell. So RNA can potentially enact replication by speeding up reactions, catalysis. So the way this would work is based on the sequence of an RNA, it can fold into a particular shape and bind these reactants and push them together to make a product in this nice movie. And there could also be roles for proteins in the earliest cells, which would be to stabilize the membrane, stabilize RNA. But there's a problem. which is that there's no known environment that's suitable for both RNA and for membranes. So for example, membranes do not tolerate certain salts well. For example, mostly +2 charged salts like magnesium. So here's some microscope pictures on the left. Get my cursor. So here on the left, we've got no magnesium, and we have these nice circles, which this is a cross section. So these are spherical membrane compartments. But when we add 10 millimolar magnesium, this completely disrupts the vesicles and the membranes, and yeah, we're not left with anything. So membranes, prefer something like fresh water, but on the other hand, RNA actually requires magnesium. So here's an example. So RNA would prefer something like salt water. So here's an example of a reaction catalyzed by RNA, and we see that it doesn't actually reach its sort of peak rate until above 20 millimolar magnesium, which is going to disrupt membranes. So what's the solution to this problem? Maybe it's carbonate lakes, because carbonate lakes have this intermediate magnesium concentration. So I know a lot of you guys in the audience are intimately familiar with these carbonate lakes. I apologize if these close-up pictures of the mud are very triggering for you. But as the name suggests, the most abundant anions in carbonate lakes are carbonates and bicarbonate. And because the solubility of magnesium carbonate is pretty low, these lakes actually maintain a relatively low concentration, as their maximum, relatively low concentration of magnesium cations. And compare that to sort of optimal lab experiments with RNA, where you're working with like 50 millimolar magnesium. So here's my hypothesis. Carbonate lakes are a Goldilocks zone for the first cells, in that there's enough magnesium for RNA, but not too much magnesium for membranes. So could the first cells have formed in carbonate lakes? Well, some work that I've done at UW recently and before the rotation shows that at least the membrane compartment can function in this lake water. So you guys may have heard me talk about this before. I'm not going to spend that much time talking about it, but basically what we're looking at in these microscope pictures that I took are clumps of many, many membrane vesicles, and they're retaining this aqueous dye. So anyway, that's good. Membranes seem to be functioning in carbonate lake water. Now, so my rotation and this talk is going to be about RNA. So in part one, I'm going to tell you guys about how I'm going to test really whether RNA genomes can be copied without enzymes in carbonate lake water. And so this this process could have allowed the earliest cells to replicate their genomic information, and this is a precursor. Replication is a precursor to evolution, which is a defining feature of life, really, I think. And then in part two, I want to ask, can RNA enzymes, ribozymes, actually function in this carbonate lake water? And so this is the role of RNA catalyzing reactions. Yeah, I'm annoyed by this bar up here. Okay, well. Beautiful, thank you. So part one of the talk is, we're testing RNA copying in carbonate lake water. So I'm just going to try and explain this process of RNA copying without enzymes. So, we start with an assumption, which is that there are random sequence RNAs that could have formed in this carbonate lake or just in the environment somewhere. And there's some reason to be skeptical about this assumption, but there's also some reason to think that this is reasonable. So then the second step is base pairing between complementary RNAs. So on the top, the smaller piece of RNA, we will call the primer, and the bottom one we'll call the template. And then free nucleotides can come in here, base pair with the template, and react with the primer to extend it by one nucleotide. The next nucleotide can come in, base pair, extend the primer, and we repeat this until we have full extension of our primer. Then we imagine some mechanism to separate these RNA strands. And so then what we've done now is to synthesize the complementary strand to our original RNA. And we could imagine doing this process again with that complementary strand to synthesize a true copy of our original RNA. So this is, as I say, this is the first step towards replicating information, which is a precursor to evolution. So the key step in this process is called primer extension. So that's what I've put into this red box. And that's what I'm going to focus on with my experiments, is the primer extension step. So here's kind of a schematic for the experiment. This is what I did. So we went to those carbonate lakes on the Caribou Plateau, took some lake water. When I got home, I filtered it. Okay, so that just removes all the cells. That's step one. In step two, I add RNA primer and template, and this dilutes the lake water just a little bit. And then I add a solution of activated nucleotides in step three. So this is where the reaction starts, okay? Now, what do I mean by activated? Normally, when we think about activated nucleotides, we're thinking about triphosphates. These are activated actually differently with a molecule called 2-aminoimidazole, which could be more likely to happen on the early Earth than having triphosphates, but it definitely reacts better. So we could talk about that more. I'm not going to spend too much time talking about that. What you need to know is at step three, the reaction starts when I add these nucleotides, and then I'll measure this reaction at various times. Now, in order to measure RNA copying, I had to learn a new technique, and that's why this was a great project for my rotation. So I showed you those microscope pictures before, and for that, I'm taking pictures of membrane vesicles, which are relatively big. So microscopy works great for those, but if I want to see extension of an RNA by a single nucleotide, microscopy is not going to get the job done. So like I said, I had to learn a new technique. And so, so I traveled to the Shostak lab at the Mass General Hospital, and I worked with some really great grad students and postdocs there. And I learned this technique, this new technique to me. It's an established technique, but this is gel electrophoresis. So basically we have a slab of gel here, and I'll put my RNA sample into one of these wells and then apply an electric current. And so the RNA will migrate towards the positive electrode because it's a negatively charged molecule. And smaller RNAs will move farther through this gel than bigger RNAs. And so then I can separate unextended RNA from RNA that's been extended by one nucleotide and RNA that's been extended by two nucleotides and so on. And I can then quantify how much of each RNA is there by fluorescence. So here are the results. So the first thing that I found is that the rate of adding the first nucleotide, so this is the reaction we're looking at, adding one nucleotide onto this primer. The rate of that reaction is actually similarly fast in lake water as it is in sort of these optimal lab conditions. And so this was surprising, and this is, I think, extremely exciting. So on this plot on the y-axis, we have the rate constants for this reaction. So we measure something for these kind of standard lab conditions. And actually the lake water, the experiments done in last chance lake water or good enough lake water, those rate constants are slightly lower, but they're not 3 orders of magnitude lower, which you might naively expect because it has much lower magnesium. And actually in the artificial lake water, which is just some salts that I mixed together to sort of mimic the ionic composition of the lake, that actually gives you an even higher rate constant. So anyway, all this is really encouraging. It's saying this reaction is really pretty similarly fast in lake water as in these lab conditions. Now, it's a slightly different story at longer times. So after 72 hours of reaction, the yield of extended primers is actually lower in lake water than in standard lab conditions. So we're looking at the fraction of all of our primers that have been extended by 3 nucleotides. That's on the y-axis. And so in standard lab conditions, that's actually pretty close to 100%. But it's a lot lower in last chance lake and good enough lake and the artificial lake water as well. So this would be a problem. I mean, if we can't actually copy our template, we can only add one nucleotide, we can't really copy our full template, that's not good. But there's a solution to that problem, which is just add more nucleotides. So the yield of extended primers is increased when we have our initial concentration of nucleotides, when we increase that initial concentration of nucleotides. So this is a similar graph to what I just showed you. Y-axis is the fraction of all primers that have been extended by 3. And so then in gray, we start with 20 millimolar of our initial nucleotide supply. And gray is what I just showed you, that data. But black is where I'm now using 100 millimolar of initial nucleotide. And I see that in last chance, in all of these like natural and artificial lakes, that yield more than doubles. And so that's great. So this is telling us, if we, maybe compared to the optimal lab conditions, it takes more nucleotides. It's less efficient in that way, but we can still, drive this reaction to very high yield just by supplying a higher concentration of nucleotides. And then something else that's interesting is that this reaction actually continues beyond 72 hours. I stopped those measurements at 72 hours, but I did a couple at longer times, and this continued reaction could improve the yields of primer extension even more. So what we see in this plot on the y-axis is fraction of primers extended by 3. And this reaction, it increases, it's starting to level off, but it increases pretty steadily for even up to 2 weeks, which is pretty impressive. When it's a 2-month project, it's hard to do reactions that last for 2 weeks. But nevertheless, this makes you think. One sort of caveat to this is after about 2 weeks, you start to see, so this is our primer, and this is some internal standard, and we see these other very faint bands forming. And so this is breakdown of that primer. Actually, you guys can't even really see it on there. It doesn't matter. It didn't happen. Just kidding. This is all, yeah, this is something to keep in mind. It's encouraging. It tells us this reaction occurs pretty well and over long times in this lake water. So that's really good. So can RNA genomes be copied non-enzymatically? Yes, they can be copied pretty well in comparison to sort of optimal lab conditions. So in part 2, I want to know, okay, can RNA enzymes function in lake water? And the answer is yes, I would say. So here we have a self-ligating ribozyme. So what does that mean? So in black, this is a piece of RNA that has assembled into a shape that allows it to be catalytic. So it's an enzyme, a ribozyme. And what this ribozyme does is it base pairs to this gray template RNA and then ligates itself. So it forms a bond between itself and this blue RNA. So you can think about this reaction as a way for, this RNA is growing itself by doing this reaction. This could have been an important step in allowing early cells to replicate their information. So I set up this reaction in 75% lake water, and I actually didn't observe any reaction after 20 hours. But when I adjusted the pH of the lake water, so I just bubbled in carbon dioxide into this lake water, bring the pH from 10 to 9, then I actually see reaction after 20 hours. And so that's encouraging. Now, you might say to me, okay, well, why do I care about this? You've changed the lake water. This is not the real lake water. This ribozyme that we have was actually generated through an evolutionary process that occurs at pH 8. So really what I've done by changing the lake water to pH 9 is make it more similar to these evolution conditions that we use to generate the ribozyme. And I think that future work could identify ribozymes that actually function optimally in unmodified lake water if we repeat that evolutionary process at, in lake conditions. So anyway, I think this is still pretty encouraging. It says that there's nothing really about the lake water that would prevent activity of these ribozymes, this result. So yeah, so I think, the upshot of this whole talk is that yes, it's plausible at least to think that the first cells could have formed in these carbonate lake environments. And that's because we have function of membrane compartments. We have we can copy RNAs, or at least extend RNA primers non-enzymatically, and RNA enzymes are able to function in this lake water. So this led me to think about sort of like a life cycle or development cycle for the first cells in carbonate lakes. So we know that these lakes have seasonal fluctuations. So in the wet season, everything that's dissolved in the water is fairly dilute. Membranes can form, they can grow, divide. I didn't talk about any of that, but it's something interesting to keep in mind. Now, it's possible during this low salt phase, if the magnesium concentration falls even lower, maybe we have reduced replication and activity of RNAs. But later on in the year, during the dry season, where the water is saturated with magnesium, that, you know, what my results show is that we can have replication and activity of RNAs. And so that is encouraging, that maybe there's kind of a, you know, a phase for membranes to do their thing, and then a phase for RNAs to do their thing. Okay, so that is all I wanted to tell you guys about. This is a very important slide because I really have to thank all of my collaborators and people from this lab where I did my rotation, the Shostak lab. I worked with some really great people, and I had a great time doing this project. I learned a lot. And also big thank you to all of my UW collaborators, and also to, you know, UWAB for you know, enabling me to do this, really, for funding this rotation. So I'm very appreciative, and yeah, happy to take any questions, chat about this more with you guys. Thank you.
Hey, questions for Zach? Let's see. Let's start with Roy.
Yes, I never asked this before, but so what is it about the composition of the lake water that sort of apparently is compensating for the low magnesium?
Right. I think it's the high pH, which makes the RNA primer more reactive. So yeah, so it's I mean, it's an interesting thing because at high pH, RNAs themselves will degrade easier, but you have low magnesium, and it compensates the hydrolysis, and it sort of works the other way for the speed of primer extension. So the pH, I think, is the answer to that question.
But you had to lower the pH for the ribozyme, right? When you bubble the CO2.
Correct, for the ribozyme, yeah.
Right. Well,
Yeah.
it's still a high pH, 9.
Yeah, yeah.
Connie?
So my question, you assume that those template RNA exists, but how is it possible to, like, well, I wanted to know more about that assumption itself, but also like how do you make those like template RNA?
Yeah, that-
Where did the nucleotides come from?
Yeah, that's a great question. It's like a major point of uncertainty for the whole field, really. But one thing that's unique about these carbonate lakes is that they can achieve very high phosphate concentrations, which is very unusual in like natural earth environments. So that's one key advantage of thinking about the carbonate lakes, is they can at least have the phosphate. There's kind of synthetic pathways that people have come up with to make nucleotides. You know, they're sort of complex. You may have like gripes with certain steps in them, but they do exist. At least we have some idea about how the nucleotides could be formed. And then actually taking nucleotides and linking them together into just the very first polymer, that can happen in kind of these seasonal dry periods. Drying out, it's a condensation reaction linking those two monomers, and so by drying out, you could form a polymer of RNA.
There's also like enzymatic activities that are template free for like RNA and also DNA, and they would be like interesting to those and so like see how to
Enzymatic activity, you said?
Yeah, there are like enzymes that build RNA without like any template. So like it would be interesting with like polymerases that are concentrated, like how they build RNA.
Yeah. Yeah, that could be an interesting model. Although what we're trying to think about specifically is before any enzymes existed, how did these first polymers get assembled and how did they get replicated? But yeah, like the way that like, yeah, Q beta or something does that, maybe it could be really informative.
Lots of questions. Sebastian.
Yeah, so very good. I've already got a few months visiting Boston.
Thank you.
I was wondering if you could remind me of, so you got those 3 nucleotide extensions. Can you remind me of like how far up you have to get for it to be applicable to your template? I seem to remember something like around 100, where you basically, maybe the question could be how many nucleotides for like a ribosome or something.
Yeah, well, for the RNA, that's a great question. Like how many, so we're adding +3, how many do you need to be able to add non-enzymatically to actually make an enzyme, for example? I think that, so this enzyme that we use, I want to say is about 75 or 80 nucleotides. So, you know, we're not there yet. We're not, I mean, so the experiments that I did, it's, you know, we're looking at a pretty simple template system where we only had 4 free bases to add to. I think, the extent of what can be done under optimal lab conditions is still only like 20s to like roughly 20 nucleotides, which I think there are some RNAs that can demonstrate some level of enzymatic activity with roughly 20 or 30 nucleotides. But in general, that is still like an unsolved part of this question, is how do we start non-enzymatically replicating long templates with, like a diverse kind of general sequence. So yeah, so I would say we still have a long way to go in answer to that question.
Yeah, follow up. So yeah, you
Oh,
were just saying.
sorry.
What nucleotide.
Did you have a question?
Sure.
Before you go, right?
Great talk, finally.
Thank you.
You had a result where, the like RNA and the primer extension started to like degrade. The solution was to add more nucleotides. How would that work in the context of like inside a membrane in
Well,
the like lake
Oh,
water?
that's a great question. So first, just to clarify, so we don't actually start seeing any degradation of the RNA until, 6 days or really like 2 weeks. So the experiments that I talked to you about where I added more nucleotides, the problem isn't that their RNAs are getting degraded. It's just that for whatever reason, they're not reacting. But anyway, how would that work inside the membrane? That's a really great question. So one thing that I wanted to try, so one thing that people know is a big molecule like the RNA primer or like the template is gonna, let's say that gets encapsulated in the membrane, it's gonna like diffuse out of the membrane relatively slowly compared to the rate that small nucleotides diffuse in. So that's a way you could kind of enable this reaction to happen in vesicles and stay inside the membrane vesicle. Yeah, that's probably the answer. Right.
Okay. Roy and then Nick.
Sorry, but just following up there, you were just using one nucleotide or something?
No, so let's see. So this is the sequence for the primer and the template. So at the end of the template, there's space for 4 C's, but again, it's just GGGG. When you start having a more complicated template, it gets harder.
Nick.
So Sebastian sort of asked my question, kind of, but I mean, I was just wondering, what is the distance, between these sorts of experiments and something like evolution? Like, is this like 5% of the way there, or is it like there are many, many problems?
So you should have solved the origin of life during your rotation. So disappointing.
No. It's a great question. I mean, I guess before I try to, yeah, that's a great question. I mean, I guess, the talk is saying we can get just as close to solving the origin of life in a test tube as we can in this lake water, which is cool, right? This is a real environment where you could imagine it happening. But yeah, how close are you to evolution? I don't know. I guess you'd probably have to be able to do this process with like a sequence general template up to 30-ish, maybe more, fill in, like extend a primer 30 bases on a template. And then, we didn't even talk about separating those strands. That's a problem. So there's still a way to go before you can just make copies like of any sort of template. But once you have that, then, all you need to do really is apply selection pressure and you're evolving.
Yeah, but the question is for what? What is the original function? And suddenly they say that too. I mean, if it's for self-replication, fine.
Yeah. It could be membrane, it could be stabilizing membranes, it could be, yeah.
Chris.
Yeah, is there a worry about deprotonation of the RNA bases due to the pH that might affect duplex formation?
That's a good question. I am not, let me see if I have, this is maybe not something we really want to, not really related to your question, but you're saying like just modifying of the base itself at pH 10?
Yeah, some of those bases have pKa's of like 9. So there's like, there might be an issue with like hydrogens coming off the bases, which might affect like how the duplexes would come together specifically.
Absolutely, yeah, that's
about that, but it seems like there's duplexes forming. So
yeah, right.
Obviously.
Yeah, but that's a great question. Maybe that, I mean, because we're only also using Well, at least, the free region of our template is only GC base pairs, but yeah, maybe for the other bases, that could be a problem. That's a great question. I don't really know the answer actually, but I'll thank you for asking. I'll look into that more.
Okay, any more questions?
Maybe.
Oh, Zoe?
So that was awesome. I recall the model system, the NPDNA is where that primer extension can go furthest, if I'm remembering correctly. Have you considered doing lake water with NPDNA, or is that, gonna be complicated for others?
I don't think it would be complicated. I think it would be doable, and I think it would be interesting. Zoe's talking about sort of a modification to the RNA really, that enables you to copy a much longer template. So that would be really interesting. Yeah, just to see how well that could work in there.
Because I think those are getting up to like +20. So they're near a lot closer, probably to fall.
Yeah.
Do you have an online
Yes,
question?
online question. Sure. It says, what roles can microenvironments play in the links for facilitating these processes?
Yeah, so I mean, I guess, when we start thinking about extremely low water activity, maybe we could have different regions of the lake that have different dry and wet states. That's a great question. I'm not exactly sure. I think there could be a role, and I think it's having microenvironments is definitely something that could happen during drying phases in the lake. And then when you mix them back together during the wet period, yeah, that could be important and maybe make some interesting chemistry happen.
Okay, well, we're out of time, so let's thank both our speakers again. Thank you, guys. And if you have more questions for Zack, then come down to the front.